In this video we will discuss about this aging CD8 plus T cells and their growth factor can affect the cognition functions negatively. Or so basically we will discuss the abstract, and in the subsequent video we will discuss the results that are presented in this figure. So what they are able to find out is that in the case of these aged mice, there is these CD8 plus T cells are formed, and these CD8 plus T cells will release this granzyme K, which is the lactic granule, and this granzyme K will thereby affect the plasticity of this neuronal and also negatively affects the synaptic changes. And because of this impact, there is a cognition decline observed in the mice, and that things they have validated by injecting the aged CD8 plus T cells into the young mice, and they are able to see a cognition decline. However, by targeting the CD8 plus T cells and also by targeting the granzyme, what they are able to find out that they are able to see the rescue in the plasticity, the related neuronal and synaptic changes, and also there is a recovery in the cognition that they have observed, and the cells behaved. And these things they have tested by injecting these anti-CD8 antibodies and anti-granzyme antibodies into the aged mice, and thereby in these conditions they are able to see a recovery in the cognition effect of the mice. So this is what this paper is talked about, and one things that people one need to understand this anti CD8 plus T cells are the cytotoxic T cells, that is the ability to release granzyme and perforin. However, why they are releasing granzyme in the aged condition, whether they are seeing any foreign particles or it is the defect of the aging CD8 plus T cells, that we will discuss in the when we will see the results that are presented in this paper. But they use this heterochronic parabiosis. So basically, if they switch these cells, they are able to they switch the CD8 plus T cells. They are able to see the changes in the mice model. In the mice, for examples, they are able to recover the cognition behavior in the aged mice, and they can see cognition decline in the young mice. I have not checked their results yet, but this is what it seems like from the abstract. And this is mainly because of this systematic environmental age. So as the CD8 plus T cells, or as the mice get aged, they are able to say that the immune system gets declined, and once the immune system get declines, it acts negatively towards their neuronal and synaptic changes, as their own immune system is trying to degrade the cognition function of the body. And this is recently published paper. So, and this impact is mainly focused since the cognition effect functions are mainly dependent on the brain. The impact is mainly focused on the brains and how these cognitions are able to recover once these anti-CD8 and anti-granzyme antibodies are used. But whether they have also shown the side effects of this, that it might prevent the immune system response against the actual foreign particles, because granzymes is the way by which the CD8 plus T cells attack the foreign particles or any virus or bacteria. So these things will also need to be considered if one needs to use this approach for actual treatment. However, this idea should be this study could provide an idea that one can target this granzyme and CD8 plus T cells through other ways, that will not only improve the cognition but also keep the immune system of the patients in an active state. So that's all for this video about the abstract. Some of these informations are from my own understanding and from my own knowledge. However, we will discuss the results in the subsequent videos that are presented in this paper, to understand how they come up to this point and what are its advantages and disadvantages. So that's all for this video. Thank you for your time.